In this video, we're going to talk about MTT assays. These assays are very simple assays that are designed to allow you to see viability and proliferation of cells. So let's start with a simple overview of how this assay works. So as I just said, it's an assay that provides a readout of cell viability and growth, and it works by measuring cell metabolic activity. So the idea is that if your cell is live and if it's growing, then it must be metabolizing. And if we measure that metabolic activity, we can indirectly say that they're alive and they're growing. So MTT is a soluble powder reagent that is added to cells and incubated with the cells. And this is very, very cheap and easy to obtain. And so this is a very popular assay to do for that reason. So this powder is added to the cells, it's incubated with the cells in regular media, and any viable or proliferating cells will contain NADPH-dependent enzymes, and these enzymes will naturally reduce MTT. It's just a side effect of their normal function. And when they reduce MTT, it will generate these insoluble crystals known as formazan. And these are purple crystals that form on the bottom of your plate. So now, after my incubation period, once these crystals have formed, if I remove the media off of my cells really carefully, and now I dissolve these crystals in DMSO, because remember they're insoluble, then I will solubilize them and I will get a purple solution. And this purple solution will change color. Its purpleness intensity will change based on how viable those cells were and how many crystals were formed. So if only very few crystals were formed and the cells are really mostly dead or not growing, then you have 0% viability. And if they are growing very well, then you sort of could call your control condition a 100% viability based on how purple it is. So this is the basic idea behind the assay. Now, of course, now that I've told you this, it's easy to see that there are some very obvious pitfalls for this assay. So the cells have to be actually metabolizing for this assay to work. And there are some cells that metabolize, but they just don't metabolize very fast, or they're not growing very fast, or they don't have the same reduction activity. And so you'll get sort of a false negative readout with your MTT. So that is something to watch out for. And there are other assays like cell titer glow, or you can get these live dead viability stains, or you can do FACSs like flow cytometry for live dead. And those are all also ways to read out viability of the cells and see how they're doing. You can do BRDU, which is sort of a growth marker, and do flow cytometry for that to assess growth. So there are many other ways to do this, but we're talking about this because it is, although it has its pitfalls, it is really simple. It is very cheap to do. You can do it in a very high throughput way. And sometimes, assuming that your cells metabolize and assuming that the conditions are met, it's actually a pretty good way to get a quick result that you can then verify with maybe one of these other methods. So now that we've talked about the assay, let's talk about the actual protocol. So it's a simple assay and the protocol is fairly simple. Basically, you would want to start by seeding your cells that you are going to do an MTT on. So you would use a 96 well plate for this usually, because you don't need so many cells to be able to see a purple MTT solution. You only need a few that are metabolizing. So usually 3,000 to 5,000 cells is sufficient. You can do even less if you have very highly proliferating cells. But the important thing is that every well must have the same number of cells, because it is affected by the number of cells. So you cannot change the number of cells unless it's actually your experimental condition to change the number of cells. MTTs also require a lot of replicates to get good error bars. So you should make sure you have like 6 to 8 replicates for each condition. And you should also make sure, as always, that you have an appropriate control, because without a control, your MTT is not going to be able to be analyzed. And then finally, before you actually add in the MTT reagent, your cells do need to attach to your plate. So if you've seeded them one day, you need to wait at least 24 hours, and then you would do any treatments that you're interested in doing, and then you would perform your MTT. So at this point, we're assuming that you've seeded the cells, you've let them attach, you've done any treatments you want to do, and you're ready to do your MTT. So at that point, you would make your MTT stock. An MTT stock should be made at this concentration, so 5 milligrams per ml in sterile PBS, because remember, you're doing all of this in the tissue culture hood and it's all sterile, so it needs to be in sterile PBS. From this, you will dilute your MTT 1 to 1,000 in appropriate culture medium for your cells. And this will give you a concentration of 5 micrograms per ml. And then you will add 110 microliters per well of MTT media solution. And it's always good to do all of this with a multi-channel. It just makes it much easier to get through the volumes and the pipetting and to be consistent. So once you've done that and you've mixed, pipetted up and down, mixed it in, you're going to incubate for 4 to 6 hours at 37 degrees. And one important point to note that I didn't make is that when you add in this 110 per well, you do need to make sure that you've already aspirated the previous media in the cell, and that ideally you've sort of aspirated by pipetting it up and down once to wash off any debris, or you've aspirated, then you've added PBS to wash, and then you've added this MTT media solution. So you're not adding the MTT media solution on top of any existing media. So once you've done that, you've replaced the media with your MTT solution. You're then going to incubate for 4 to 6 hours, sort of in your standard TC incubator. So all of this has to have been sterile. So you have to make sure that everything you do is in the TC hood up until the readout point. Once you've incubated for those 4 to 6 hours, you're going to carefully remove the media with a pipette. So here, do not aspirate. If you aspirate, you will remove all of the crystals at the bottom. So instead, pipette very gently and pipette up and down once so that you sort of wash any debris, and then remove. And then add 100 microliters of DMSO per well. And you're going to pipette this really well to mix it with the crystals, because now you want the crystals to come into solution. So you want to pipette very aggressively to get all the crystals to come into solution. Whereas here, you could even not pipette up and down once if you were worried. You really don't want to disturb the crystals at that point. So if you just carefully remove everything, that is sufficient. You don't want to mess with the crystals there. But here you do want to mess with them. Here you want to get as strong of a pipetting as you can. And then you can incubate. Some people say you can incubate 10 minutes at room temperature. Others say that you want things to get really into solutions. You should wait 24 hours and put it overnight in the incubator. I personally, for most cells, find that 10 minutes at room temp is more than sufficient. But if you feel like you're not getting good solubilization and you want to wait, you can wait up to 24 hours and put it back in the incubator. So again, you must be sterile to put it back into the incubator, because you don't want to put anything non-sterile into the incubator with all your cells. And then finally, you will read out the results. And so you'll use a microplate reader. It reads an absorbance of 550 to 600 nanometers. And then you analyze your plate, which we'll talk about in a second. So make sure this makes sense. It's a simple protocol, but there are some key points that are important to keep in mind. So one of those key points is your plate design. And so a good way to design your plate is to keep all of your replicates going down. So since a plate has 8 wells going down, this gives you 8x replicates, which is good to have. And then each column will be a condition. So these could be increasing numbers of cells. These could also be increasing or decreasing amounts of drugs, because you want to do a dose response and see how much drug it takes to kill off cells. If it is a drug, it's important to do these in a log scale. So you don't want to do like 10, 20, 30. Instead, you want to do something that jumps a little, so it's tight around the range that you're expecting, and then sort of loosens up as you go up. So for example, if I thought that TMZ killed cells at 10, then maybe my scheme would be something like 10, 5, 2, 1, 20, 50, 100, 500, and so something like this, where it's not exactly going in a sequential linear order. It's more like it's doubling or 2.5ing every single time I move up. You also want to be sure to include the appropriate positive and negative controls. And so your positive control in a drug situation would be that there is no drug. So 0 nanomolar of drug. Because in the presence of no drug, you would expect that your cells would grow totally normally and have 100% viability. And your negative control would be, I didn't even put cells in this, it's just DMSO without any cells. And obviously that should have like 0% viability. There should be no growth or no crystal formation there. So that's sort of your background or negative control subtraction. So you want to make sure you have both of those controls. And then you have 10 wells left to do whatever range you would like to do. And then of course you can do multiple plates if you need to. And so you can always extend your range up or do multiple cell lines or multiple drugs, whatever you need to do. But anything that is a control for a plate or anything that's going to be compared on one graph should ideally be in one plate. And then some other key considerations to keep in mind is that, as we said, everything should be done in a sterile TC hood until the reading step, because you might put it back in the incubator and it needs to be sterile the entire time. You should use a multi-channel pipette. I find that just makes it much easier to be consistent and to pipette the amount that needs to be pipetted. Be very careful with your crystals when you're removing media, but then suspend very thoroughly when you solubilize. And think very carefully about your plate design before you seed your cells. So before you do even the first step where you're seeding cells into a plate, make sure you've thought about the design and how many wells you're going to need and how many plates you're going to need. Because once you seed those cells and you do the treatments, you're not really going to be able to go back and add any more replicates or add any more conditions. So be careful here. And always seed exactly the same number of cells in every single well, because this assay is very affected by the number of cells. So this here is an example, and you can see that as the cell number goes up, we have an increase in our absorbance. And so if you plated some cells around 15K and some cells around 3K, it would look like there was a difference in viability, but actually there's just a difference in the cell number. So the cell number has to be the same. The only time when that does not hold is if your experiment is actually to look at how the absorbance changes based on cell number. In that case, of course, you could change the cell number. And then make sure you read it at the correct absorbance setting. Microplate readers offer a range of different absorbances, and you want to make sure you have the right one to get actual results. And then very quickly, just a quick hint, the easiest way, in my opinion, to get the same number of cells is to count them very carefully and then make sort of a master mix of cells for your plate. So if you are adding, let's say, 3,000 cells to every single well, and you know that you have 96 wells to pipette, then you might make enough for 100 wells, right? Because we talked about how you always make extra. And you would just make 3,000 times 100, so you'd make 300K cells in this solution, plus 200 microliters times 100, which is approximately 20 ml. And you would just make the solution, mix it really, really well in a tube, and then pipette out 200 microliters into every well using your multi-channel. And that way, all your wells get the exact same amount of cells. Everything is totally controlled, And so then when you do your MTT, you don't have to be worried that maybe I messed up my cell count, or maybe one of these wells doesn't have enough cells and that's why it's off. You know that you're going to get exactly the same number of cells across the board. So now let's talk about analysis. So your plate reader is going to give you an output that is an absorbance measure. And once you have that, this is the formula you're going to use. So you're going to subtract out for every experimental well, you're going to subtract out the mean of the negative control, or the background. So this is your DMSO only, right? So it should have no readout. But in reality, it usually shows something around like 0.04 or 0.00 something. So you want to subtract out any background. And then you're going to divide by the mean of the positive control. And that essentially means that you're setting your positive control at a 100% viability. Because again, just like qPCR, this is a relative measure. So you are deciding that there is something that is considered your 100% or your control. And then you're measuring viability and proliferation over that control. So then you multiply by 100 to convert it to percent, and you end up with a percent viability measure. So to make that concept a little clearer, I wanted to walk through an example. So let's say that you ran a plate and this was your MTT data. And obviously in reality, you would have 8x replicates, not just 4, and you'd probably have more conditions, but this is just a pretend plate. This would be our positive control, so no treatment, and this would be our negative control. And you can see that this would probably end up being this pale pink color, and this would be bright purple when we look at our plates. And then all of our treatments range somewhere in between, so you can see that our treatments are slowly killing off our cells. So to analyze this, let's say we start with this number. We want to analyze this number and say convert it to a percent viability. We're going to first calculate the averages of our controls. So this is the average of all of the positive controls, and this is the average of all of the negative controls. And then we're going to take this 0.8945 and we're going to subtract out this background. So it's going to be 0.89 minus 0.04. And that's going to give us something like 0.85. So that's this top part. And then we're going to divide by the mean of the positive control, which is 0.87. So we're basically doing 0.84 over 0.87. And that gives us 96.65 once we multiply by 100. So this is about 96% viable. So make sure that calculation makes sense. And then once it does, you can just do this in Excel. And it's very easy to put in the formulas and just extend it across your whole table. And then you will get all of the different viability percentages. For the no cells, you can see that our numbers are very strange. It's negative and positive. And that's because, of course, we don't expect this to actually show any viability. So it doesn't really surprise us that the numbers are kind of weird. And this is something that we would never show on a graph either, because the numbers are kind of weird, and because it's really just meant to be a background subtraction. You're saying that in the absence of cells, this is what the assay still reads out, so I have to take that out. And then I have to divide by my 100% control in order to get a percent viability. So you can think of this like how we subtract out GAP-DH or how we always compare to actin. You're just sort of taking out the basic background that's on every single sample. And then you're calculating the rest. So as we've said, this formula will give a viability for every control well that should average to 100 because you're normalizing against the control's average. It will also give you a percent viability for every experimental well. And then you would take all of these replicates, so this stack of 4, you would take this and put it into Prism, and that would generate your graphs with error bars. And typically we show this on a line plot, so it will look something like these are your percent viabilities here, and then these are your doses, and this one would probably look like, let's see, 0 has like a 90% viability, and 30 has like a 30% viability, so probably something like that would be what your graph look like. And then typically, as I said, we show the positive control, but we will not show the negative control because it is just a background subtraction. But you have to show the positive control to show that you have a control. Otherwise, everyone's first question will be, well, What did you normalize against? Where's the control? Like, how do we know where your 100% is? So that's the basic analysis. And then remember, your conditions don't have to be drugs. They can be time. You could do like, I did 10 micromolar drug concentration and I looked at it at 1, 2, 3, and 4 days. You could do cell number. You could do different types of drugs. You could do drugs and combinations of drugs. So there's really no end to what you can do with this. It's whatever condition you want, as long as there's an appropriate control. So now that you have those replicates and you've put them into PRISM, let's talk briefly about presentation interpretation. So for presentation, all the usual things apply. You want to have clear labels, clear error bars, clear explanations of anything on your plots. And like I said, we usually show this as a line graph with percent viability on the y-axis and then an x-axis of our drug conditions. And usually if it is treatments, it's in a log scale. So it's sort of that like 1, 2, 5, 10, where you're like constantly doubling rather than being a linear scale. So here, this one is one example of an MTT assay. And in this, they did drugs and then drug combinations. And you can kind of see how the curve shifts. And then usually the thing of interest is the IC50. So that is where 50% of the cells are dead. And so here they've calculated their IC50, and that's what they're interested in. And IC50s can tell you something about how strong drugs are or what they're doing. And so that's usually the number that people go after. And then here you can see that this person has actually varied the number of cells per well. And so they're showing how well cells proliferate in cultures based on cells per well, all treated with this drug for a certain number of time. So again, you can kind of see absorbance and how the cells look. So 2 different ways to do it. One is showing cell death, one is showing growth, but both are reasonable analyses of an MTT. All right, so I would like to end with some example experiments to think about. So for the first one, what dose of TMZ should I use for experiments on PDX lines? This is a very relevant question, right? Because we use TMZ for our experiments all the time when we want to know what TMZ is doing to the cells. And one way to choose this would be to just look into the literature for what dose human cells are probably experiencing in the brain. But that would be a little bit hard to find. You might find some estimates, but it would be hard to pin down exactly what dose of TMZ every human cell sees. So maybe you just want to know, what's the IC50 of TMZ? What's a good place where I'm stressing the cells enough that 50, 60% of them are dying, but 50% of them are surviving, and maybe I can see what factors play into resistance or something like that. So you want to know a reasonable dose. So to do this experiment, you might design an MTT plate, right, with a range of doses. And like we talked about, you would have a positive control here, which has 0 dose, and a negative control here, which just has no cells. And then here you might range from like 0.1 to 100 of TMZ. And then this would be your 8x replicates. And then perhaps you'd have a plate for every cell line you were interested in. So maybe you could do 43s and 6s and 5s and U251s. Those might be cells that you're interested in looking at. And then you would analyze this and you would be able to generate an IC50 for every line. And that IC50 would tell you how sensitive every line is to TMZ. So 43s have a really high IC50. That says that it takes a lot of drug to kill 43s, and maybe they're really resistant to TMZ, whereas maybe 5s are really sensitive and they die really easily. So it is useful to know for every cell line what the TMZ effect is. And then the second one I'll leave for you as practice, but you could imagine that you would compare a knockout and a control sample and graph that out to see how cell proliferation might change. So this is some real data from a paper that's referenced here. And you can see that different cell lines have different IC50s. So for example, this GBM12, the IC50 of where half the cells are dead is maybe somewhere around here. Whereas for these others, they don't hit their IC50 until like somewhere over here. So we'd probably say that GBM12 is more sensitive to TMZ than any of these other cell lines. These are more resistant because even at high doses of TMZ, they're not really dying very much. And so we could probably treat some of these cells with even 140 micromolar of TMZ if we wanted to. But if we did that with GBM12, we'd end up with only 20% viable cells. And so that would be a problem. So this would help you pick a reasonable dose for all your cell lines, or help you pick a reasonable dose that is cell line dependent. And then you can see that they've compared and done the statistics for all the different cell lines, which is also very good to have. So that's a quick overview of MTTs, both how they work and how they should be analyzed and presented. I hope that that was helpful. If you have any comments or questions, you can feel free to contact me or contact the lab. Thank you so much for listening, and feel free to subscribe to our channel to see more similar content.